Welcome. This webinar is brought to you by the New Hampshire Training Institute on Addictive Disorders. You can access the recording of this webinar on our website at www.nhadaca.org within our webinar library. After completing this webinar, please complete the quiz and evaluation if you're interested in receiving a certificate. You can find the quiz and evaluation on our website next to the Watch Now button you selected to view this webinar. If you're a member of NHADAC or a NADAC, the certificate for this webinar is free. If you are not a member, the fee is $15. You will receive a certificate worth 1.5 continuing education credits once you have completed and passed the quiz and paid for the webinar if applicable. Through the New Hampshire Training Institute on Addictive Disorders, this 1.5-hour event is pre-approved by the New Hampshire Board of Licensing for alcohol and other drug use professionals, as well as NBCC. If you have any trouble viewing this webinar, have audio problems, or have any technical questions, please contact us at 603-225-7060. Please email any questions regarding the material you see today to training@nhadaca.org.
So before we dive into the actual the research that we do in our lab, because a lot of what I talked so far is research done in so many different labs across the world in the United States. In my lab, we don't do research on rodents. I don't have mice and rats in the lab. So I'll talk about what we do in my lab in the next section. But I do wanted to kind of give a few slides on tolerance and dependence because people confuse these different terminology, and I wanted to just kind of clarify certain things. So when so tolerance is basically when you reach a stage where the organism, the individual, no longer responds to a drug, and actually that happens relatively fast when you're using morphine for pain management, and then you need to have more morphine and more morphine. So it's in that aspect. It just does not, the body's not responding to the effect of the drug itself, so the original effect. So if we're talking about the analgesic effect, now you need a higher dose to achieve that analgesia, to ease the pain of that patient. So that's called tolerance, okay? So tolerance, so it has to do with the analgesic effect of the tolerance for this analgesic effect happens in the thalamus, and I'm pretty sure I talked about the thalamus earlier. It's this big egg-shaped region in the center of the brain, and back then at the beginning of this discussion, I was talking about the thalamus and pain signals that go through the spinal cord and arrive in the thalamus, and then these pain signals are sent to the, do you remember what I said about this? What's this area?
So it's prefrontal cortex?
Prefrontal, frontal and prefrontal are here.
Yeah.
Frontal cortex is here. This is decision making.
Right.
So what would be here?
The sensory?
Correct, sensory.
Yeah.
Yes, and that's the sensory is in the parietal lobe, so it's behind the frontal lobe. So frontal lobe goes all the way to here, and you have movement over here, and then right behind it in the parietal lobe you have sensory, and everything lands there. And we have like there's a big gyrus that goes from side to side, and depending on where the sensory information comes from, it lands on this side of the brain, or on this side, or on this side. There's a really cool map about where sensory information lands, depending if it comes from your mouth or your hand or your back, it's really cool. So it arrives there, and that's where it's interpreted. So the thalamus is redirecting this information, this pain signals, to the sensory region of the brain for interpretation. So what would be great is to cancel these signals to the thalamus, and that's why the analgesic effect of opioids are on the thalamus. So they suppress the signaling going through the thalamus, so there's no there's no pain signals. That's what it is. There's no pain signals going through. Well, reduction of pain signals going through. Now, when you reach a state of dependence, this is when the individual only functions normally if the drug is present. So in a state of tolerance, you get the morphine, and now you've taken it for a while now, and it's really not doing much to your pain anymore, but there is no more morphine available, so you're just living with the pain, but but although your other bodily functions are still normal. In this case, your whole body is going to react very badly if you do not have the drug given to you. So that's when we reach the level of dependence, is a state where you only function normally if the drug is available. So we have a physical disturbance at that point when the drug is not there. So this is what we call the withdrawal effect. When the drug isn't stopped being given, now you have all these other side effects that take place. Not just, I just feel the pain, but it's not getting any better. I just still feel it. If I can't get the morphine, higher morphine, now I just feel more the pain. That was tolerance, but now if there's more side effects and physical disturbance, then you have this sense of withdrawal. So the dependence itself develops in the thalamus, but is more linked to the brainstem. I just wanted to kind of clarify this terminology for these two words, just in case not everybody had that. And I'm just adding 2 slides. You are all probably more, because you're on the front line, you're more aware of all the common symptoms of, for example, here, heroin withdrawal. So I personally know individuals that went through substance use disorders, and a very dear friend of mine, and it stemmed from actually having Crohn's disease all his life, and the pain from this condition led to using fentanyl at high doses, and then trying to cut back down. He had a really, really hard time, and it's just basically now he's better. But I remember during his withdrawal phase, it was an entire week of just very, very, very difficult changes, body changes. The nausea was terrible, he told me, and really pain all over your body, fever. So it's really hard, really hard. And it takes a while, it's not in 24 hours and you're better. It really is a weak process, at least. So some of the symptoms can last longer, and when I go through my research, I will actually show you why we believe that relapse can take place even if the drug has been stopped and the person is in recovery and it's been a year, and sometimes there is relapse. So I'm going to actually show you at the molecular level why are we having such high relapse rate? Because some individuals have to go through I'm losing the word now. rehab several times, and they get better and they relapse, and then they go back to rehab, and so it's very tricky. Okay, so this is just my summary slide for really where this is going on in the brain. So we talked about the reward pathway and how addiction is affecting the reward pathway, right? This is the reward pathway: VTA, nucleus accumbens, and prefrontal cortex, right? This is prefrontal cortex, where there's some of the decision making and so right here. And then dependence has more to do with the thalamus and the brainstem. Okay, so why do we use opioids? What are the physical effects of opioids? So let's summarize what we learned here. What's the number one reason we use opioids?
The release of dopamine.
As clinicians, why do we use opioids? I'm sorry, I meant
For
to.
pain.
Yes.
Yeah, pain management. Yeah.
For pain management, correct. Yes, so I saw Darren also type it. Is there a neurophysiological basis for post-acute withdrawal? Well, we're going to mention that. And if I don't answer that question by the end, remind me again of that. So yes, it provides pain relief, okay? So this is really the original purpose of prescribing opioids, right? I was talking about from the point of view of a clinician, what's the physical effect of opioids and why we use them is to provide pain relief. What's the second effect of opioids that, is, unintentional and really why it ends up being something that we get addicted to? It may be too simple of a question.
The euphoric effect and ending of like kind of the mental suffering, escapism.
Right. So it's in the beginning, this is really the second effect. So I'm telling you, I have athletes on campus. They student athletes, they've gone through, they go through such rigorous workouts and schedule. They're injured all the time. I see students coming in my classroom on crutches and where they have surgery, ACL surgery, and they're so young and they already have tooth surgeries and they're on opioids. And we talk about that openly in the classroom because a lot of these addictions start with an initial problem, an initial injury, an initial surgery. I remember when I had my wisdom teeth out, my dentist prescribed an opioid. When I had, my children, C-sections, they prescribed opioids. So 15 years ago, that was more. Now it's, I don't know. Actually, I don't know if they still prescribe opioids after delivery, but definitely was a bit looser of how they gave opioids back then. But the effect is that calming effect. I wouldn't say the euphoria as you think of euphoria, like you're going crazy. I don't know if some people think of euphoria like that. The euphoria meant here is a very, very, you feel lighter, you feel calm, you feel warmth. I added the sun here for warmth. So it just gives you that warmth throughout your body, and it's a very it's a good feeling that comes with that. And that's the problem, is that feeling of warmth and lightness and calm and that good feeling that's the feeling that's very addictive. You're like the rat on the lever. It's like, oh, I want to feel that again. I want to feel that again. It's just really, so initially that's how it starts. So, but then because of the dependence, you feel like if you do not have the drug, then you know you're going to feel bad. So you're looking for the drug to not feel bad, as opposed to, you know, they talk about chasing that initial feeling. It's really not the case once you are really addicted. And the third, I should have asked that question, and I moved the slide a bit too fast. I wanted you to tell me what was that third impact, because we didn't talk about that yet, and that is a very big problem. So I want to share that story from John. A client described heroin like being cradled in my mother's arms. Those are the descriptions I heard as well. It's just a warmth feeling, an undescribable good warmth feeling. So the problem is, the third effect is that we have opioid receptors in the parts of the brainstem that are responsible for respiratory. The respiratory center happens to be in the brainstem as well. So this is the brainstem that's a bit more detailed than the one I showed you before, right? VTA would be somewhere here. This is the midbrain, pons, and this is the medulla over here. But across the medulla and the pons, we have these different respiratory centers. And these respiratory centers have opioid receptors. So when opioids are in the system, they're going to bind there as well. You cannot, well, this is actually one of the direction of some of the research is how can we make them bind to in the thalamus and prevent, you know, be as analgesics and use that effect and prevent the binding on the receptors in the brainstem. We're trying to figure that out. How can we kind of push the opioids to just bind up there and not in the brainstem? Problem is that all of these receptors are the same. So you can't, for example, knock out the receptor and then now you don't have it anywhere. And, you know, it's not, that would be the answer. But so for now, it's a problem because the respiratory center gets depressed. The respiratory center is depressed. We say depressed, and your breathing is shallower and shallower, basically fall asleep and do not wake up anymore. You die in your sleep. So it's definitely not a violent death, but it's very scary. So there's many stories of Narcan saving people's lives, and this is just one story of Miss Hunt, I believe her name was. No, Hurt. Her name is Hurt, and she willingly participated in this Narcan call this not advertisement, a campaign to bring awareness. That's what I was trying to look
Public
for.
service announcement.
Yes, to bring awareness to Narcan and how important it is, how it saves lives, and the way our Narcan works. I can kind of like, you know, draw this real quick here. So you have the receptor here, and you have the opioid that's going to bind on here. And what Narcan does, this is Narcan, it bumps the opioid out and sits on the receptor. And it's so strong, it literally like cuts it off like right away. As soon as it's in the system, it bumps it off and sits on it and basically blocks it because it doesn't have the downstream effects of, it doesn't have the euphoric effects and the analgesic effect. It just has an effect of being an antagonist. It just pushes the opioid off and binds on the receptor and says, that's it, I'm here, I'm not moving, and that's it. and the individual literally wakes up. Sometimes it takes 2 or 3 doses of Narcan, but the person wakes up. Sometimes they're upset because they lost that opportunity of being in that stage, but their life is saved. So I'm gonna, before I move to my research, what's the time? 10:30. Oh, good. I'm looking at the chat. Can you speak a little about spin drives when people stop using for a while to get that sense of ease again, and how most of the time they OD after this amount of time of waiting? Yes, this is a good point. Is this correct? I know a lot of people go into detox to get away for a bit, to have that strong high when they get out. Oh, wow, that's really tricky. So some people go to detox just so to go back to honestly, I didn't know the expression spin dries, but I understand what you're referring to. Here, I have some space here to draw something. So on the surface of a cell, you have these opioid receptors, right? I'm going to call them mu opioid receptor. These are the most important. Mu. This is the Greek letter mu. Mu opioid receptor. There's a beta and there's a delta. So mu opioid receptors are really important for opioid binding. Opioid binding. And then you end up with the more usage there is, then the cell goes, oh, I need more receptors. I need more receptors. So there's a more opioids binding originally, right? So the cell adapts to the opioid being present and creates more receptors. Unfortunately, tolerance builds up as well, and you can have all the receptors possible on the surface, and you're not going to have that increase of the feeling of that high. So when you go into detox, the cell realizes there are no more opioids available. So then it goes back down to just being, I'm exaggerating here, to just being back down to one receptor. So when you expose that cell again, the effect of just all of these opioids all of a sudden is actually going to be stronger than all of these receptors there that reached a tolerance level. So the buildup takes place and the high gets higher, right? The more receptors, the more we have that effect. But there's a reach of tolerance, and to kind of get rid of that tolerance, it seems like some people have used that. I didn't know. I didn't know that people go into detox to basically get back to their original level. But so at the molecular level, this is the tentative explanation. You're basically tweaking the number of receptors on your cell and how the cell responds. So it's really incredible. I walked in on someone who used, and their breathing was super shallow. This is from Christina. They were starting to turn a gray color, and I shook them awake, and they slowly started to come back. And then the color came back, and no Narcan had to be used. Oh my God! So these are the stories that you have encountered and you have experienced, and I've never experienced something like that. But that's just incredible. So you can shake them awake. There is a point of no return where the Narcan would have to be administered. So in that particular example, it seems like the point of no return wasn't reached, and you were able to just wake them up. There was just enough of the respiratory center where enough oxygen was reflushing the tissues. That's what it is, right? The respiratory center means that we're, moving the lungs so we can have oxygen, so we can have the circulation of oxygen throughout our body. And that's where, the color changes and from, pale to back to having a normal skin color. I've been told by every patient over the years that they chase that feeling from first high, but never can get it again. Right, exactly. So I was, so I don't know about so maybe the detox story is, some individuals discover that if they detox and then come back, then they get that, that high again. But if there is a long-term use, it's more about trying to keep your body in a normal state and not have these, physical withdrawal symptoms that are very hard, the sweating, the fever, and the nausea and all of that. So yes, they use just to not have these bad symptoms, and they really never actually feel the high again. It's just very sad. It's a disorder of the brain. It's literally, I'm explaining to you today that it's at the molecular level and has nothing to do with, willpower, and this whole notion is I think we've moved on from that that bad perception, and people are getting more knowledgeable on all of that. So John wants me to explain again, when more receptors are present and the using stops, the receptors go away. How quickly does this happen? I'm not sure. I'm not sure of that. It's not overnight.
I didn't put a question mark. And using stops, the receptors go away, question mark. And how quickly, but it does, they just kind of go away. They
yes,
they just stop, they recede, or
yes, they are actually dismantled. It's like little Lego pieces. It's amazing. The cell builds these receptors from amino acids. Amino acids, you have that in your meat and your the food that you eat. The proteins from all of your meat is broken down into these little, they're called monomers. These are amino acids, and these amino acids help build proteins. So your cells get the nutrients from your diet, and then all of these little monomers then rebuild the proteins that the cell needs. So these receptors are proteins that the cell will build if we need more, and will dismantle if we don't need. And it's a very dynamic process. Now, time-wise, I'm not too sure about, it doesn't take overnight to have all the receptors change. And that's why there's that time frame of detox. So I don't have numbers on time frame, but it literally dismantles it or rebuilds it according to need. Yes. Okay. Okay. So let's talk about my research. All right, so I think by this time you have a really good understanding of not only the normal reward pathway and how it works at the molecular level, and maybe that helps you understand how it's really directly linked to the actual happenings in the brain at the cellular level, at the molecular level, and how opioids can disrupt that and create a havoc on different parts of the brain. So your decision making, yes, of course, it takes pain away, but then the decision making is affected, and the respiratory center is affected, and that those are the consequences. So I arrived at Franklin Pierce in 2013 from another position as a professor somewhere else. And in 2013, this is when I arrived, you have a year to kind of settle down and think about what kind of research you're going to establish. I was just establishing a brand new research project. And at the time, there was really a lot in the news about the opioid epidemic and the talk about overdoses and deaths and all of that. And all along, even before that, I was always interested in opioids, the effect of opioids, because I personally knew very dear people to me that were affected. None of them died, thankfully, but it really was, it affected me. So it was a personal interest of mine. So I decided I want to study this. And so I'm showing you this national graphs to show you overall in the country. It was really bad right around that time, right? So right after I arrived at Franklin Pierce. And New Hampshire, where Franklin Pierce is, it was ranked, and it's still ranked, I checked that, second in the nation for, those are per 100,000 people. So because of, it's per 100,000. So the number of deaths per 100,000 citizens, we ranked numbers 2. So this is a map showing you where the highest rate, West Virginia, 40 deaths per 100,000. New Hampshire was 32, Ohio 31, Maryland 30, Massachusetts 29. So these were the highest ranked states. So, and at the time, well, it's still now, the New Hampshire Medical Examiner, there were only 2 because it's a small state, and they were overwhelmed. They were really overwhelmed. So I'm like, this is good. So for funding, this was great because there was an interest in the opioid crisis. So I was able to obtain funding because, of course, funding is related to, what's really important and the questions that need to be answered. So at the time, let me go back here. So at the time I arrived, I literally had no lab. So it was really starting from scratch. And there is no animal facility here. So normally research is really usually done with, animals and rodents and mice. And so I thought, okay, I don't have an animal facility. What kind of research am I going to do? And I thought, maybe I can just do research on tissues, and I can do, these types of techniques in the lab are more straightforward and require less of a setup than an animal facility. So I thought I'm going to look at human brains. And at the time, I don't know if I was, over optimistic, but it was really a long shot to think that I'm just going to go to the New Hampshire Medical Examiner and say, hey, I want to look at those brains that you are, dissecting out of, these individuals during autopsy. And it actually took me 11 months to convince Dr. Thomas at the time to let me participate and collaborate and research on those tissues. So I was very, very proud. I was very happy I got to that point. When I talk to colleagues in other states, they tell me it's absolutely incredible. There's no way a examiner is going to, there's so many examiners in other states that wouldn't do that. And so I think this was to the credit of the New Hampshire Medical Examiner. They were open-minded and realizing that this is really important work. And basically all of my trials at trying to explain how important that was paid off, because I was able to convince them that this is really important, and we need information from humans. And this is what was lacking, information from humans. So at the time, so let's concentrate just on this. I didn't make this into a I should have made it appearing one at a time. So try not to read the rest. So you probably learned the scientific method in school, because I know it's explained in middle school, and then it's reviewed even at the college level in biology. The scientific method is literally applied even at a researcher level. This is how we think as researchers. We make an observation, and from all the readings I did on the topic, right? You know, when you're trying to establish a research project, you read what other people have done first. And at the time, I read that a specific molecule called delta phosB, this is my baby. You're going to hear a lot about delta phosB in the next hour. It was described as the molecular switch for addiction in the rodent model. And this is Dr. Nestler, who wrote countless number of papers about that, and he used to work on depression. Now he works on depression and addiction, and so many fellows, postdoctoral fellows from his lab are now their own researchers. He's a really big name, and he's the one I approached at one of the conferences, and I told him about my collection, my collaboration with the New Hampshire Medical Examiner, and he was really shocked, and he was like, let's collaborate. So he is actually my collaborator on this project, and I'm happy because he's a big name. He knows what he's doing, and I get tremendous feedback from him. So I'm really happy that I'm collaborating with him. So it's in his labs that it was discovered delta phosB is a very important molecule that kind of creates this cascade of events that is really detrimental for addiction. And my question, very simple, because it wasn't done at the time, is this molecule present in humans? All the work I was reading is mice, rats, no humans. And I'm like, this is it. So that was my research question. My hypothesis was, yes, it's in humans. I bet you there is a molecule in humans that has that same, role, importance in humans. And that's my quest. I'm going to go look for that delta phosB in humans. So my experiment, which is what I proposed to my funding committee, I'm going to test human brain specimens and going to go look for delta phosB and, prove that it's there or not. And if it's not there, then we rethink the experiment, and we, have a new hypothesis and do something else. So this is what I'm going to talk about next. So if it gets a little bit too technical, stop me, don't hesitate, or unmute yourself if I don't catch a chat, or if it's too much, just, maybe give me a sign on your end. Oh, so I am going to show some brain specimens, and I wasn't sure of everybody's background, and I don't want to offend anybody. If you do not want to see brain specimens, I just want to give you a heads up that I'm going to talk about that. So you can maybe also text. Well, it's on a lot of this next slides. So I don't have any problems from a minute, or if you can maybe just, or shut your screen or something. But I'm not showing people or autopsies. I'm showing just those brain specimens that I received. So this is what I brought to the medical examiner. I brought jars, and I stood there during autopsy. While I participated in many autopsies, even when I was at Case Western in Cleveland, Ohio, I was already collaborating with volunteering at the medical examiner there when I was a graduate student, working on my PhD. So I was always fascinated with medical examiners. I'm telling you, there's so many fascinating careers in life that, you could take so many different paths, and they're all really cool. Or forensic investigation, or all of the stuff I'm interested in. But so I brought these jars, and I said, when you have an overdose I would need the middle section of the brain, because I wasn't asking for the moon. I really just wanted the center, because that's where nucleus accumbens is. It's in the center, and that's what I wanted to test. I want to see if delta phos-B is in nucleus accumbens in the center of the brain. So I asked for just that center of the brain, and I got a really big chunk, and I was just so happy about that. So I brought this one from the lab to show you. I don't know. Yes, you see that there. So this is how they came in these jars. So then they would call me. They would say, "We have your specimen. It's available." Like right at autopsy. So the next day, they will put in 10% formalin as a fixative, because we don't want the brain to disintegrate, decompose, we say. So there's a fixative, and that's what was in the jar that I provided. And then I would go pick them up and bring them back to Franklin Pierce to dissect them in the lab, which is the first picture of me dissecting one of those. So I obtained from the medical examiner a number of opioid cases, 17, so that's 24. Yeah. And some were females, most of them were males. The average age was, around 35, so really young. And I did obtain control cases from individuals that went through autopsy for other reasons. And because I needed control cases to compare what I was going to show in my opioid cases with the control cases. And because I was not able to obtain enough control cases, I'm actually going to the brain bank to supplement these control tissues because in science and research and for publication purposes and all the people criticizing your work, critiquing, I should say critiquing, not criticizing, that has a negative effect. Critiquing, meaning that, how can you make it better? You need more controls. So I was told, you need more controls, you need more controls. So I'm going to get controls from brain banks. Brain banks is an incredible resource of brain tissue. People donate their brain for research or for Alzheimer, for all kinds of different, reasons. There's a brain bank in near Boston, just focused on CTE. It's a chronic traumatic encephalopathy. It's the brain disease that happens following concussion in boxers and football players and things like that. So the brain banks are very, very useful for researchers, and I'm in the process of obtaining more control from brain banks, age matched and sex matched. So there will be chosen not an 80-year-old Alzheimer brain, but more of a 30-year-old. And then you look at the history of what they passed, how they passed, and then you will try to match it as much as possible with your own specimens. So when I dissected the brain, so you have to have a good knowledge of the anatomy of the brain, and I would, find all of these different regions that I would be very interested in, and I would dissect them out. So I dissected the amygdala, which I haven't talked about at all, except one time when I talked about teens. The amygdala regulates the fear, that interprets the fear that is presented to you, the danger situations that presented to you, and if you're going to be fearful or not fearful about it. And this is actually an important part of addiction that is now important in research because, I'll get to that. And then hippocampus is that region in your brain, and I mentioned that earlier, that is involved in memory storage, memory retrieval. So formation of memories is all in the hippocampus. So of course, when I saw the hippocampus, I would be getting very excited. Oh, this is the hippocampus. This is so amazing. And then I would just, dissect it out and save it. And all of these parts, I put them into these cassettes that, so they're just kind of sectioned out. And this is the hippocampus, and this is corpus callosum, midbrain, da da da. And I would just put these little pieces, parts in all of these cassettes, so we can test hippocampus versus nucleus accumbens versus amygdala. And then, so this is really technical. I'm just showing you behind the scenes in a research lab working on opioids. I hope this is something that you'll find interesting. So then these little cassettes, they add little slits here. When I sent them, you see how there are these little slits here, and there's just this little piece of tissue in here, the specimen. And then, but again, they get dipped into wax. And then you have a wax block. A wax block is called paraffin. And why we do that? Because it's much easier to cut a block of wax than to cut the tissue directly with a slicer. And the slicer is called a microtome. This is a microtome. So we have histologists that are, very skilled at doing that. This is another career that's very rewarding, to just work on working on the tissues. This is what histology is the study of tissues. And so that block is kind of like sliced very, very thin, like 5 micrometers thin slices. And then you get one of these slices and you put it on a glass slide, just like that. So this is the tissue that's now on a glass slide, and then we can do analysis. This is my student Annie, right here. She now graduated, but oh, she was so amazing. She was so good. So now we take these glass slides, and you see her with these forceps. We do different baths of different alcohol baths here to de-wax them. It's a whole process to de-wax them, to then do the experiments. So I'm very lucky. I have this beautiful collection. It's like of course it's not a brain bank. It's like a mini collection that I have with I dissected 414 blocks so far, with different sections of different parts of different brains of different individuals. It's very, very precious. I really am very honored to have that and to be able to do research on that, to help advance our knowledge in the field. So this is our lab. We even use a pressure cooker in the lab. It's one of the steps in the experiment. Yeah. So let's get back to why am I interested in delta phosphate? And hopefully you'll find this really interesting, because it's going to answer the question about the relapse. This is what I want to get at, about the relapse. So as I mentioned, Dr. Nestler has published many manuscripts about that. And one of them, so now this is like 20 years ago, we understood 20 years ago that delta phosphate is a sustained molecular switch that establishes the state of addiction. Literally, it's like the holy grail that everything happens downstream after that. So that was established back then, and so it's still the case. So we know this is true. This is work that was published and published by other labs since then. So this is it. This is really an important molecule. And bear with me. I'm going to be a little bit technical about the molecule itself, but hopefully you'll see why I'm doing that.
Can you go back to the last slide just for 2 seconds?
Yes, of course.
Thank you. Okay, thank you. I'm all set.
Okay, so delta phosphate. So since we discovered it was really important, there are other articles that have established that, why is it important? Because it alters the expression of other genes. Do you know where genes are? Let's just try to evaluate where we're at. When I talk about genes,
Inside the cells.
inside the cells, inside the nucleus, it's part of the chromosomes. It's the DNA. So you have a DNA molecule, which maybe you're familiar with this helix look to it. This is the DNA. The DNA is basically where all the codes are, the codes for all different kinds of molecules, collagen, right? If your skin needs to regenerate and make new collagen protein in the skin, there's a code for collagen here, and then there's a code for, opioid receptor here. These are proteins that are, the code to make the proteins is in here. So think of the DNA as your cookbook that includes all the recipes to make the product, and the final product is the protein. This is the final product. So if you have a molecule that's going to mess up the gene, and you're not going to have the normal or appropriate level of downstream proteins, that's a problem. And we discovered that delta phosphate, this is what it's doing. It's messing up the expression of other genes, and it's just creating this cascade of events that leads to addiction. So this was published in 2016, responsible for establishing long-lasting changes. Delta phosphate, when it appears in your tissue, in your nervous tissue, it stays there for a long time. This is the problem with following abuse when you're trying to be in recovery, why there's a relapse. And I'm going to explain to you why does it stay so long. So this was part of Nestler's work. So actually I did have, I forgot I had this here. I had this diagram prepared ahead of time to remind you of, if you have never seen this before in the biology course, how in the cell, so this is the cell, like you were saying, you said that it was inside the cell. This is your cell. Inside the cell, there's a compartment called the nucleus. This is where the genetic information, this is the genetic information. The DNA is your genetic information. It basically houses all the codes for everything. And every gene is basically the code for a particular molecule. So you have one gene that's going to be, we say transcribed, but it's okay if that's not a word you're familiar with, into an intermediary molecule that's called RNA, and then it's going to go out, and then we're going to have the protein made. So this is a summary of how we get from the genetic information, the gene, in the DNA molecule, this is the DNA molecule, and we end up with one protein, okay? One gene, one protein, and of course, now you understand the cell has so many different proteins it needs to function normally. It needs receptors, it needs transporters to recycle things. Remember the reuptake of dopamine back into the cell, those were transporters or receptors to accept neurotransmitters. So we have all kinds of proteins that are important for the life of a cell. So delta phosphate is a protein. And delta phosphate stems from phosphate gene, the gene, right? Remember I said here, there's a gene for every protein. So there's a phosphate gene. And the phosphate gene codes for a phosphate protein. And this phosphate protein has a specific size. This is the size. It's okay if you don't understand this is the number of amino acids. Remember the monomers? I told you amino acids string together to form a long protein. 338 amino acids, and this is the weight of it. And this FOSB protein is a normal protein that you have in your brain. You have it. You have it in your brain, and it regulates. Here, I'm going to go back to this. It regulates this. So normal FOSB oops, I think my computer is going through some kind of background refresh and it's lagging here. FOSB regulates this process. And it's normal. We need that. Remember when we talked about receptors? Oh, we need more receptors now because we have more opioids. Or, oh, opioids are gone, so we need less receptors, so we destroy them. If we need more receptors, there's going to be a regulation of, let's get this gene decoded so we can have more receptors. Remember when I told you the cell will just make more receptors, or will destroy the receptors, or make more receptors. So we go back to the gene, the recipe, and we will make that receptor and kind of embed it into the membrane. So FOSB is a regulator of all kinds of things to be made. That's what I wanted to say here. But when you're under a stress or a drug abuse, actually I shouldn't draw all this because I have these arrows. There is what we call a splicing effect, and we don't have to worry about really what that means. In the end, it's a shorter form of FOSB, and you have a delta FOSB protein that is truncated. Truncated means it's like shorter. It's only 273 amino acids, so it's a little bit smaller in size. This is the size. And it's very stable. Why? Because the portion that was spliced out, splicing means cutting, the portion that was spliced out is the portion that's important for its recycling. So FOSB protein actually has a region, like let's say this, to help it get recycled, which is good because then it's like, you know, when it's not needed, it goes away and it gets recycled and da-da-da. But the splicing event takes this away. And now we have a protein that does not have a portion that targets it for recycling. So it stays there. That's not good because it really disrupts the normal. It's supposed to be recycled and come back and get recycled and come back. So if it's stable, it actually is going to disrupt things. And not only that, it gets even more stable with the phosphorylation, as an additional phosphorylation step. So this was, you know, these are all different experiments that were done to establish all this knowledge. So this delta FOSB gets phosphorylated and now it's really stable. It is not going away. And so it acts as a regulator just like the regular FOSB did. So with another molecule, it comes, they're called transcription factors. Maybe you've heard that in the past. And they land on genes here ahead of them. This is like a helicopter pad. It lands there. And then it's going to stimulate or not stimulate. So it's going to prevent the decoding of these genes. So all of these downstream, these genes, these are example of genes that are affected by this new protein here. And all of this leads to addictive behavior. And all these are references of publications that worked on that. So when you have a deregulation of these specific CRAB and BDNF and CDK5, these are genes that are important for normal functioning. And if they're deregulated, you end up with addictive behavior. And we, in the mouse model, we proved that delta FOSB deregulates these genes. So my aim for my research was to look at a delta FOSB in humans by immunohistochemistry. So I started by a very simple question. Very, very simple. Is it there or is it not there? Let's start by just at least looking at it. Because remember, all the publications that I'm mentioning back here, all these publications here and this is all on mice and rats. So I want to see if it's in humans, and that's what myself and my students are doing in the lab. We're doing a lot of immunohistochemistry, which we're looking at delta phosphate in the tissue slice. Remember how I told you this? We slice the tissue very, very thin. And then another aim that we have is analyze the differential gene expression between opioids and non-opioid cases. So we want to see, and this is really exciting because this is the first time we're looking at all the genes that are expressed in an opioid brain versus a person that has not taken opioids. And we're comparing what are all the genes that are just like really highly regulated or completely down versus, you know, I want to say normal brain, but the challenge with humans is that, you know, that there's so many variables. It's not like a mouse that you just keep in a cage with the same partners and the same food and no other variables. So it's very, very challenging to work with human tissue and make sure to compare what's comparable. But I won't talk about that aim because it's just going to get a bit too heavy on the research discussion. But I can tell you about what I visually saw. So just a really quick, and then you'll have a knowledge on immunohistochemistry. I think this is kind of good to know. It's like a advanced knowledge on the technique itself. So I'm looking for delta phosphate. Is it there or is it not there? That's what I'm looking for. So it's a protein in the tissue, or maybe it's not in the tissue, right? So I'm going to do all these dots to represent delta phosphate. And the way we go about this is we use an antibody that's anti-delta phosphate. Antibodies are part of the immune system. They're meant to go and recognize antigens, which usually are the pathogens, so we can kind of like neutralize them. This is part of your immune system. And an antibody here is used solely for the purpose of research and technique, because it's going to go recognize something we want to recognize, we want to find out. It's kind of like a bait and switch method here. And these antibodies, anti-delta phosphate, they're commercially available. The pharmaceutical companies that sell you these things, they literally inject delta phosphate in a rabbit. The rabbit reacts to the delta phosphate protein. Goes, what's this? What's this? The immune system kicks in. Now you have antibodies that are against delta phosphate. They harvest that, and now they sell the antibodies to researchers like me. Here, you want antibodies? It's going to cost you $300 for a tube with 100 microliter, 1/10 of a milliliter. $300. Side note. So I don't know if I lost you, but I hope not. Just relax and just kind of like get a feel for what it is on the research side. So the first antibody is added. So my student or myself, we add with a pipetter, we add a liquid on top of the tissue. And then we have to add another layer with a second antibody, because the second antibody has a specific enzyme that's going to allow us to put a substrate. And then we see a brown precipitate, and then we can actually see something, because these tissues are actually translucent. When you slice them and you have them on the glass slide, they're actually not colored like here. They're translucent. You can't see anything. You put on them the microscope, it's all going to be clear. And doing this scaffolding of antibodies with we call this a colorimetric assay, where you end up with a color. It allows you to see. And then, we do washes. We wash this between steps. So if there was no delta phosphate, what would happen if delta phosphate wasn't in the tissue? What do you think would happen here in my case?
There would be
You
no
wouldn't
brown
get-
precipitate.
There would be no brown precipitate, exactly, because there are washing steps. So whatever didn't bind, right? If there's no and delta phosphate, I need, this has to be washed off. So whatever bound stays and whatever didn't bind washes away. So if there's no delta phosphate, all of this is going to wash away, which means the second antibody is basically going to be useless, but that's okay. That's part of the process. But it's going to get washed away because it can't bind on something that got washed away from the step before. So if there is no delta phosphate to begin with, you do not see a brown precipitate. We add a second dye. We use a second color. We use blue. A blue dye at the end to see the other cells, because if you have cells that are brown, it's a bit kind of complicated for me to change colors in my pen, but if you see cells that are going to be brown, the other cells are going to be clear. And in order to see all the cells that are even negative, they don't have delta phosphate in them, we added a blue dye in them. So we focus on 3 regions, and now you're all familiar with that. This is the reward circuit, the reward pathway that involves the VTA, nucleus accumbens, and you see how nucleus accumbens communicates with the prefrontal cortex, and there's actually communication with the hippocampus. Oh, I forget I have all of these fancy, I'm drawing, but I have all these fancy things. Nucleus accumbens, hippocampus, and amygdala. Those are the 3 regions that I focus on because they're very important in addiction. When they say an alcoholic, if they go back into a bar, maybe that's going to be really hard for them because they're actually going to, it's going to trigger something in their brain because they're remembering a setting that was that they liked and they were addicted to it at one point. So hippocampus has a very big influence on addiction. And amygdala, we're understanding more and more about amygdala and its involvement in addiction. So nucleus accumbens, this is just the map of a brain to show you, it's right here. So in my dissections, this is, the whole striatum. And nucleus accumbens is called the ventral striatum because it's at the bottom. And when I would see it like that, oh my gosh, I would get so excited because I was, because it's actually in a very narrow area of the brain. So think of it as like a pointy bowl. The striatum is a pointy bowl, and the nucleus accumbens is at the bottom. So it's really only in a one like layer, one thickness, if it's very thin here. And then if you kind of slice over, then you lost it. And if you slice on the other side, you lost it. So when I would see it, I would get really, really excited. Right, and then it goes in the cassette and etc. And this is how it looks like when it's on a slide. So this is from my lab, and this is how it looks like on the slide. So you have a whole section here on the slide, and it's very clear, right? You can't see anything. You put it on the microscope, it's very hard to see the cells. So it's very important because so I already told you that it involves targeting the genes. And this is what I saw. So you have all these brown dots, and then you have these blue cells. Of course, this is a very, very low magnification. And this is one of our opioid cases. We had a very strong delta phosphate protein expression in most of our opioid cases. And when you zoom in at a higher magnification, you actually can see the neurons better, right? Better than the previous magnification. Some opioid cases did not have as many brown cells, right? Every time you see a brown cell, that's where delta phosphate was or is. And the brown precipitate is basically telling you here, this is here, delta phosphate is right here, and here, but not in these blue cells. You see all of these other blue cells, these are negative cells. They do not have delta phosphate in them. So in some slices, some regions. Well, here we only focus on nucleus accumbens. We had very strong staining across the board. And then we counted the cells and we compared with blue cells versus brown cells. And we had like 73% positive cells versus some cases only had 44% positive cells. So we did some analysis like that for some kind of quantitative process. And so now we're getting even more up close. So this is a higher magnification of neurons. Look how beautiful this is. These are neurons you see there. And then, you know, those, see these stellates, the dendrites, they're kind of buried under. You don't see them all necessarily, but you catch one here and then another one here. So here are neurons right up close and personal. This is one of my favorite slides. And these are negative cells. So we tested 3 regions. So the second region was the hippocampus. So I want to show you where the part of your brain that stores the memory, and this is important in addiction, because with research, so this is a reference of a research done in the rodent model in rats, delta phos-B disrupts the dentate gyrus, which is a very specific region in the brain, in the hippocampus. So maybe this is a bit too technical for you, but it's in the hippocampus. So it disrupts the hippocampus. We found that in the rodent model, the rats. And this is where we have neurogenesis going on normally and formation of new memories normally. So that gets disrupted with addiction. So following chronic drug use, we have inhibition of neurogenesis. So there's not a replenishment. Neurogenesis means we have new neurons made and replaced. And so we're inhibiting that. Development of drug-seeking behavior linked to the hippocampus. That's why I'm interested in the hippocampus, because we want to understand what's going on. Memory retention, information processing, and problem solving is impaired following chronic drug use in the hippocampus. So that's why it's important to look at hippocampus. So here's hippocampus. This is following my dissection. So the hippocampus looks like a seahorse. That's why we call it hippo, like the seahorse. So this is the original specimen, then it's embedded in wax. This is the wax here. And this is the slices on the slide. So I'm kind of showing you, reminding you of what the process was, the dissection, the embedding, the slicing. And literally, I mean, one of my slides looked exactly like the drawing of a hippocampus from a seahorse. And I just thought I would add this next to it to show you. It's literally, it looks so similar. It's such a beautiful region in the brain, and it was so important. So every part of it is important, but hippocampus, this is what degenerates in Alzheimer, which is terrible. Okay, and what did we find? Because I'm sharing some of my research results here, which is not yet published, so this is top secret. But we're working on the manuscript to publish this data. It's unpublished data yet, so anyway, so yes, so look at all this brown staining in very specific regions of the hippocampus. They're called CA regions, CA1, CA3, CA. So this is, we don't need to go into that, but very specific. So this is meaningful. One, we proved that delta phos-B is in hippocampus, human hippocampus. So I'm just showing you some slides here of just different pictures of hippocampus. So this is all from my lab, just different parts of the hippocampus, the dentate gyrus from different cases. I believe this is not the same case as this one. This is from another part of the hippocampus, the CA regions. Look at these neurons, look at them. Isn't it amazing that you can see it in a microscope? You can actually see the cells that are doing this electrical signaling between each other. They're connected to each other, and they're talking to each other, and it's just really amazing. And every region has its own purpose of what other region they're connected to, and where do we go from here? Do we go talk to the frontal cortex? Do we go talk to the amygdala? All of these connections are back and forth. So beautiful. I'm just kind of showing off all these slides here. I just want to show you the negative cells versus the positive cells. So the staining process works. My students are really good. My research assistants are really good at doing that. Yeah, so we have positive cells and we have negative cells, so which means we have expression of delta phosphate in these particular cases. All right, and the final region I want to share with you is the amygdala. Why am I interested in the amygdala? Because again, according to research, this was done in other labs. Amygdala is implicated in mediating signs of withdrawal in subjects with either acute or chronic opioid exposure. So the negative, anything that is negative, the negative effect from the addiction condition is related to the amygdala. The amygdala is that very small almond-shaped region in your brain that regulates fear, perception of fear. I told you, if you remember, teens might not be as fearful as us. I grew up in France, and I remember at 18, we went in the catacombs in France, and it wasn't legal to go under and go into the catacombs. It's like an underground system of tunnels, and there's skeletons down there, and there's a museum for it, but there's also an entranceway that only crazy teens like us go in the night and access them. And now I think back about it, and I'm like, this was crazy, because we went through some tunnels that were very, very tight. I remember crawling under a passage where I had the wall. I was just crawling in a passage where the ceiling was on my back. And I'm thinking now, like, this is crazy. What were we thinking? It could have collapsed. We could have been buried, but it was exciting. We were in the catacombs of Paris, because I grew up in Paris, and it's crazy. And I just fear for my own teens and what kind of crazy stuff they're going to do. And unfortunately, it's normal. It's part of growing up and the brain developing, and the amygdala has a different way of assessing fear and situations at different ages. So when opioids are present, it's part of that negative withdrawal response. They suppress fear behavior by modulating the amygdala. This was published, and we know now it's the amygdala that basically is telling you, no, just go for it. Let's go for it. I might die. I might fall asleep and not wake up. Nah, just go for it. Go for it. And this is what the amygdala, the signals from the amygdala are sending to your frontal cortex, because this is where the decision making is. We call it the motivational brain, when you decide to do something. So that's why I based the three regions, why I wanted to focus on three regions, because there's published research that indicates that they're important for addiction. Hippocampus, amygdala. And of course, you already understood why nucleus accumbens was important. Okay, so in some of our cases, the amygdala actually didn't show positive cells, and in some other cases, all the cells were all the cells were brown. They're pretty brown. So in the amygdala, the nuclei are much rounder, right? Every part of the brain looks a bit different. So very good question from John. Did you find that there was significant reduction of delta phosphate in the controls, the non-addicted brain? Yes. Yes, we did. What we were surprised is to find delta phosphate in the control to begin with. I don't know. I naively thought it was going to be black and white. You know, we're going to have delta phosphate here and no delta phosphate here, and this is going to be great. But actually, we were finding delta phosphate in the control brains, and we were like, oh my gosh, what did we do wrong? And actually, it is there. We all have a low level of delta phosphate because of normal stress. Remember in my diagram showing you what creates the formation of delta phosphate was drug abuse and stress. So actually stress can lead to, you know, compulsive behavior, which is what addiction is. So I don't have exact percentages, but we did compare the two and there was a difference. Yes. So I don't know if this is a bit too much now in the amygdala. I'm going to show you some slides about the amygdala, but there's subregions in the amygdala, and one is this region, and we believe that delta phosphate is concentrated in that particular subregion of the amygdala. It's called the lateral nucleus because we just saw it, and comparing it to, you know, maps of the brain, we're like, what is this? Why is it looking like this? And then when we compared it to the map of the brain, we're like, oh, maybe because that's where it's impacting the most. The delta phosphate shows up in the lateral nucleus of the amygdala because that's where it really has an impact the most. And then we're going to have to figure out why, but at least we're pinpointing things down. So what we know about the lateral nucleus from research in the mice is the sensory interface of the amygdala in fear conditioning. So when you see something that's dangerous and it was dangerous to you, it conditioned you to that. So that's what fear conditioning is. So next time you see the object, even before anything happens, you know it's dangerous, right? So that's what fear conditioning is. And so maybe I'm just kind of I'm just excited about to talk about all this, but maybe here it's involved in actually inhibiting that fear conditioning where you know it's going to be dangerous, but hey, let's just do it again, right? Because normally your amygdala allows you to evaluate scenarios that are dangerous for you, but then you are, and so you don't do them again. So you don't put yourself in those situations again. I'm thinking about fear conditioning, like, you know, when you it's so unfortunate, but you know, I tell my daughters, you know, be careful when you're a parking lot alone, make sure it's a buddy system, you're always walking with somebody. You know, they're still young, I don't want them to drive late at night alone in the car because in case the car breaks. All of this, unfortunately, it adds, you know, it's unfortunate that we have to tell our girls to be so careful. You know, more I'm going to worry more about my daughters than my son almost. But so it's unfortunately it's kind of that we're conditioning them to that. So, you know, because I want them to take every precaution possible, but it's probably also creating some fear when you are walking alone in a parking lot at night. You know, is it you know, you think of your safe or not safe, and you know, some women will walk with their, you know, keys in the hand, just ready for. So that's what I'm talking about. Is this kind of conditioning, and it can be inhibited when you're using drugs, and then you're not scared. So we were excited to see this lateral nucleus of the amygdala, but we're still not sure this is meaningful, this particular subregion, but at least we're going to describe them as these are the region lighting up in different individuals. These are different individuals. Another one here. So we're just kind of like really interested in this. So we identified this tentative pattern of expression of delta phos-B in this nucleus, in this region of the amygdala, that's the sensory interface for fear conditioning. All right. So I tested all these regions for gene expression, differential gene expression. I think I'm going to like skip that only because it might just be too much. We basically looked at all the genes. There were 770, and we just compared them between the two. And we're currently analyzing the results. And there are some candidate genes that are popping up that are really out there. They're affected in these brains. They just show up as being really affected. So let's get into the conclusion, since we only have 10 minutes left. I don't want to say more about that second part of the research, but let's get into the conclusion. I have some conclusion slides to see if you've retained anything. And if you have any questions at the end, I'm sure I will have another 5 minutes at the end. So let's do this. Let's see what you retained. Oh, this is going to be easy, just from hearing most of you talk already. Which is most potent, heroin, fentanyl, or carfentanil?
Carfentanil.
Yes, yes. And let's see, written responses and verbal. Thank you very much. So this would be carfentanil here. Yes, and then heroin is the least. Can you imagine now that we're saying heroin is the least, and fentanyl? So this is what I had shown you in that slide. And then addiction. What's addiction? This is a long. So we learned about addiction and tolerance and dependence. I want you to understand the differences between these two. Right?
Addiction is when an organism has to have it, like the behavior is altered.
Yes, yes, definitely. And thank you, Sarah, for also answering that. And Barbara is saying we now have fentanyl strips along with Narcan in office. For what? Tell me. Why fentanyl strips?
Harm reduction, we have those too. We have little packets with diluted water and then fentanyl strips and instructions on how to use them, so that when people obtain drugs, they can identify if it has fentanyl in it to reduce the harm in overdose.
So fentanyl strips to test them to see if fentanyl was in their system?
No, if it's in the drug they're about to use, it's used in harm reduction to try and keep people alive even while they're actively using.
You have fentanyl. I still don't understand.
It allows the drug user to test his drug to see if it's fentanyl or heroin, so they don't overdose, so they don't take too much, so they're more careful.
I see. Oh, this is amazing. I didn't even know that existed.
There's a whole harm reduction movement across the country, and it's very stigmatized, but there's a lot of work within it to reduce the amount of active users who have who aren't spreading HIV or hepatitis C. So they have clean syringes, Narcan's included in those kits, as well as diluted water and fentanyl test strips. And it's all secure, and it's mostly voluntary, with people who just go out in their community and find active users and give them the clean supplies and ability to survive their active addiction, so that they feel comfortable in reaching out to someone, and eventually it can lead to recovery.
Of course, I support that. This is really good. I didn't even know this was this. So it's called harm reduction campaign. Is that what you said?
Yeah, harm reduction. There's the state has an HIV and HEP resource center, And they primarily conduct a lot of the harm reduction movement and outreach within the state of New Hampshire in different counties. But there's also a force that's going forward with bills within the legislature trying to get more harm reduction movements throughout the state.
Yeah.
So sorry, just out of curiosity, where mostly everything is fentanyl now and not heroin, how is that being utilized?
So it just mostly allows the user to identify their drug, and then it's up to them whether they still want to use it, or just be aware that it's there and use smaller amounts so they don't overdose.
Okay. Thank you.
So the fentanyl strips, do they have a specific names? Darren is asking.
I don't remember. I have a kit. I have kits in my office, and I'm connected with the Ryan Fowler is a big contact within the HIV Hep resource center within New Hampshire, and he is my contact for receiving kits to reach out to active addicts to give them out and be a safety zone for them. So if anyone is interested, I'm happy to put my email in the chat, and that I could definitely get more information, or if anyone wants to become more of a active person within the harm reduction community, I can connect you with Ryan Fowler as well.
You can buy the strips at any drugstore. If you just go into the drugstore, tell them you want fentanyl strips, but you can get them for free from a lot of the harm reduction agencies. So it just, wherever you can, and there's also videos on TV, on YouTube showing you how to use them, you know, what to do and when to use them, etc. But just go on to the state harm reduction site and it'll tell you anything you need to know.
That's very, very informative. That's very good.
I put the website in the chat. I volunteer for the coalition in New Hampshire, if you're based in New Hampshire, but that's their website, and it has all the exchange and how to access the kits through New Hampshire.
Harmreduction.org. That's really good. Okay, thank you. I'm in support of anything that can help. I know other countries have even, or I'm not sure if it's the United States, but I know in Europe they have sites for, you know, safe use where they provide needles and everything for using, but safely. So I think this presentation definitely gave you a good dose of neuron communication and how they communicate with each other. This sounds so simple now that you've gone through this. You know which transmitter triggers a sense of pleasure, right? Yep, I see an answer already. Dopamine, John. Thank you for sharing your information, Stephanie. And you now are aware that there's 3 effects of opioids that they have on the organism, right? Pain relief and the, you know, the sense of, they call it euphoria, but euphoria sounds like an exciting state, but it's more of a very comforting state with opioids. And then unfortunately, the depression of the respiratory centers in the brain. This is really because the receptors are there, right? So I wanted to explain to you that at the molecular level, so you can really also convey that to people that are still thinking this is just a not having the will, but people are getting more knowledgeable about that now. Do you remember what could be a contributing, I'm a little typo here, to opioids' long-lasting grip and the theory behind the relapse? What did I say during the, it's really like
Delta Phos B.
Yes, thank you. Thank you for remembering my molecule, Delta Phos B, correct. Well, why is delta phosphate different from the normal phosphate and making it long lasting?
It's more stable, so it doesn't get broken down as easily, and it hangs around much longer.
Exactly, exactly. So literally you still have it in your brain tissue, even though you stopped using the drugs. And in their animal model, in rats and mice, we have, they sacrifice them and then they go measure and they go look for delta phosphate in their brain. Weeks and months later, it's still there. It's still having an impact on things in the brain. So it's really really delta phosphate is the problem. Okay. Yes, and I think now you're familiar with the reward circuit, that it includes the nucleus accumbens. We definitely talked about that a lot today. The amygdala with the sphere conditioning, and the hippocampus with the memory formation. So you're definitely good now about that. And I just wanted to end with just thanking my team and I'm just here with students that are mostly undergraduates. I don't even have a doctoral PhD student here because we don't have access to graduate students. But they're amazing. They're very motivated, and they're amazing, and they work really hard. And I want to also acknowledge, of course, all the collaborators I work with. The current chief medical examiner, famous Dr. Nestler, and his postdoc is Dr. Barreau, which I actually just came back from Strasbourg with a collaboration with him in Strasbourg. I was in Strasbourg last year to collaborate on the project with him in his own lab there. Now he's a big name in France. Yeah, and the shared resources I use, of course, across these different places. So, and then I have references if you're more interested in